Hi everyone, this is M.B.B.S nerd, and in this video I'll be talking about hairy cell leukemia. So let's begin. Hairy cell leukemia is a very rare but a distinctive neoplasm consisting of 2% of leukemias. It predominantly affects middle-aged white males, and the cell of origin is a post-germinal center memory B cell. Now, what is the pathogenesis? In more than 90% cases, there are point mutations resulting in increased activation of the serine threonine protein kinase. And what is the point mutation? There is valine to glutamate substitution at residue 600. 600 is basically the location at which the mutation is taking place in the protein, and this protein kinase lies immediately downstream of RAS in M.B.B.S signaling cascade. So let's visualize this. If you see that in a normal cell, when a growth factor binds a growth factor receptor, there is activation of this RAS protein, which I have highlighted here, and this RAS protein leads to increase, you can say, activation of this RAF or the serine threonine kinase, and then finally this MAP kinase leads to the uh, expression of MYC protein and cyclin D protein. As you know, they are responsible for increased cellular proliferation and growth. So due to this mutations, what happens is that this serine threonine protein kinase remains, you can say, constitutively activated, or it is activated all the time without any stimulus. It does not require the stimulus of the growth factor. Due to this mutation, the protein which comes out to be behaves like this. So that is why these cells become leukemic, and they continuously multiply and rapidly produce proteins and undergo have a pro growth kind of metabolism all the time, and that is why you see all that symptoms. Now if I talk about the clinical features, then they are seen as a result of bone marrow, liver and splenic infiltration. Hepatomegaly is rare, but generally bone marrow and splenic infiltration is commonly seen. Now there will be pancytopenia due to marrow infiltration. And why so? Because if you increasingly infiltrate the marrow, then you will compete with the other hematopoietic stem cells and starve them out for nutrients, and this will lead to pancytopenia. And splenomegaly is often massive, and this is very commonly seen. This is very important. Now if I talk about that patient generally presents with the symptoms due to cytopenia, and that will be purpura, obviously due to low platelet counts, anemia due to less red blood cells, and infections due to decrease in neutrophils, basophils and eosinophils. Generally these infections are caused by a typical mycobacterium. If I talk about the morphology, then in peripheral blood smear, we see leukemic cells with hairy projections. But why? You see, due to increased serine threonine kinase activation of that protein, increased activation, this leads to the increased activation of the cytoskeleton, the actin cytoskeleton, which is present in the lymphocytes, and this leads to increased membrane projections or ruffles which are created here. Generally, cytoskeleton is responsible for the mobility of cells in lymphocytes or the neutrophils, which are actively mobile. But due to this increased activation of serine threonine kinase, the cytoskeleton is constantly, it is trying to cause the cell membrane to change its shape and be very much mobile, or causing the cell to be very much mobile. That is why we see a lot of membrane ruffles, and that is why these leukemic cells are highly infiltrative. Also, in bone marrow biopsy, we see a diffuse interstitial infiltrate of cells with round, just like this round nuclei, oblong or reniform nuclei. Now, what do you mean by oblong? Oblong means the nuclei are oval shaped. And if I talk about reniform, then Reni stands for kidney, this keyword. Then reniform nuclei means the nuclei is kidney shaped, like this. And blue cytoplasm. Generally, we don't take a bone marrow aspiration because we get a dry tap. And why so? Because these cells are enmeshed in an extracellular matrix of reticulin fibers. And why so? Because just like I told you that due to heavy marrow infiltration, a lot of hematopoietic stem cells are dying, and due to death or cell death, what happens is that there is a process of inflammation occurring there, and if inflammation occurs repeatedly, then this leads to fibrosis. As you know, chronic inflammation, there is tissue repair and healing by fibrosis. So what happens in fibrosis? There is increased laydown of extracellular matrix. And what is the extracellular matrix in bone marrow? Yes, you are right, that is the reticulin fibers. That is why there is increased deposition of reticulin fibers leading to dry tap. Now if I talk about the gross image, then you see a large spleen with, you can say, a very reddish appearance. So the splenic red pulp is heavily infiltrated, giving a beefy red gross appearance, and this leads to, you can say, the massive splenomegaly. But why the enlargement of the red pulp of the spleen? Like in our earlier videos of lymphoma leukemia, we always saw a whitish nodular pattern, but here we see a reddish appearance of the spleen. So that is because You see, first of all, the answer for this is given in the immunohistochemistry markers, and that is annexin A1. Annexin A1 is expressed, or is a cellular marker expressed by apoptotic cells, which help in their phagocytosis, because macrophages recognize this marker and they actively phagocytose apoptotic cells. So this marker is shown by the leukemic cells, and as they reach the spleen or the splenic red pulp, in which there is a huge amount of macrophages, then these macrophages start actively phagocytosing these leukemic cells. And since these leukemic cells obviously are huge in number, they are increasingly proliferating, then there is compensatory hypertrophy of this red pulp, and that is why it is very much apparent, and this leads to the splenomegaly we see here. And if I talk about the immunohistochemistry markers, then it is positive for CD19, CD20, and these three markers you may remember or you may not remember, but annexin A1 is very much important. So I end my video today. If you like this video, give a thumbs up, comment down the comment section, and please subscribe. I will see you in the next video. Till then, bye bye.